This is my lab, biomedical optics, and the research focus of my research lab is to develop devices or techniques to detect tissue injuries or disease development in a non-destructive way. And most of the devices and the techniques developed from our research lab are optical based. So this is just a cartoon to demonstrate how the research project is developed and carried out in my research lab. And my research team concentrates efforts in two areas: instrumentation development and signal acquisition and analysis. And most of the research projects produce prototype systems, and then we test those systems in vivo environments. And this is a part we collaborate with our clinical collaborators. And from the in vivo evaluation results, and we understand the performance of the system as well as get the user experience. And use that as a feedback. We redesign or refine the system design, and so this cycle would continue until the final design meets the design specs as well as user expectation. And then next, I'm going to use two ongoing projects to demonstrate the research activities within my lab. And the first project is intraoperative brain tumor demarcation project. And here we develop a system, and it's capable to detect a residual tumor at the resection front during the operation. So the purpose of developing such a system is to assist to allow neurosurgeons to achieve more complete brain tumor resection. In the last spring, we completed a validation trial, a small-scale validation trial to demonstrate the accuracy of the system. And currently, we're seeking industrial partners to help us commercialize this particular system. The next project will be the in vivo study of epileptic cortex. And this is a collaboration with Dr. Jorge Riera, who's also a BME faculty member, and Nicholas Children's Hospital. And we developed an imaging system to study the hemodynamic characteristics of epileptic cortex. And through the study, we have identified an interesting intrinsic properties. And currently, we are using this property to develop a new approach to identify epileptic cortex intraoperatively, and that should aid epilepsy surgery. And we also hope through this study we can gain better understanding how the epileptic seizure occurs. Okay. So this is my brief presentation, and if you're interested in knowing more about my research activities, and again, feel free to contact me.